Hi, this tutorial is the first one in the biochemistry part of your A-level chemistry about proteins and DNA, and this one is an introduction to amino acids. We've got a picture of one down here. So amino acids have an amino group, so an amine end. They have a carboxyl group, so carboxylic acid end, and they have an R group which can change, and depending on what that R group is, tells you which amino acid you're looking at. So first of all, you need to know how to name amino acids, and you'll be pleased to know that they follow the normal UPAC nomenclature rules, so it should be nice and easy for you to pick up on how to name them. But being part of our biochemistry and the building blocks of our proteins, biologists have already named them. So they do have trivial names or classical names. So this one here is known as alanine, this one is leucine, this one is lysine, and there are about 20 different amino acids that are named like this. If in your exam you are asked to name an amino acid, you will be expected to name it using UPAC nomenclature. That is the systematic naming rules. So the reason I've put these in, alanine, leucine and lysine, is because sometimes in an exam they might ask you to look at this compound, it is alanine, or look at this compound, it is valine or leucine, tyrosine. Don't be put off by the trivial names. You don't need to worry about it. They will either draw the structure or they'll give you a systematic name to work with. So you do not need to know these names, but you may see them in your exam. So to name an amino acid, you need to do the normal thing that you would do, which is look for the longest carbon chain. And you are looking for the longest carbon chain that includes the carboxylic acid group, because this takes priority. So the carbon of the carboxylic acid group is going to be carbon number 1, and you count from there. So we've got 1, 2, 3 carbon atoms in this. If you're not clear on that, it might be worth you drawing this out in full displayed formula, which will help you to practice where your carbons and your hydrogens go anyway. But we've got 1 carbon there, 1 carbon there, and 1 carbon there. So this is going to be a propanoic acid chain. Okay, so we've got propanoic because it's 3 carbons, and we've got acid, which is this part here. And we don't need to say that it's 1 propanoic acid. We know that this is going to be carbon number 1 anyway, because acids take priority. Coming off of this propanoic acid chain, we've got 1 substituent, which is here, and this is an amine group. So when an amine takes a prefix, we call it amino, and we need to say where that amine group is. So this is carbon 1, this is carbon 2, so this is a 2-amino group, and that's going to go on as a prefix. So you can see now the systematic name for alanine is 2-amino propanoic acid, because you've got propanoic acid chain with 3 carbons in it, and coming off carbon number 2, you've got an amino group, or an amine group. So we do the same very quickly with leucine and lysine, just to get used to how you name them, and then that should be fairly straightforward. So in leucine, the carbon chain that has got the carboxylic acid in it is 1, 2, 3, 4, 5 carbons long, so it's going to be based on pentanoic acid. And then coming off of carbon number 1, 2, coming off carbon 2, we've got an amine group, so again it's going to be 2-amino. Okay, so that covers the amine group, but we've also got, coming off of carbon number 1, 2, 3, 4, we've also got a methyl group as well. So we've got 4-methyl. So these are your 2 prefix substituents, and you just need to make sure that you put them in place in front of the name pentanoic acid, but you need to make sure you get them the right way around. And remember, it's alphabetical order, so the A and the M, you need to check that they are in alphabetical order, so that's fine as it is. So it's going to be 2-amino, 4-methyl, pentanoic acid. Okay, so finally looking at lysine, we have got a carbon chain including a carboxylic acid of 1, 2, 3, 4, 5, 6 carbons, because remember this bond is going to a nitrogen, so just 6 carbons up to here. So it's going to be based on hexanoic acid. Okay, and in terms of substituents coming off this main carbon chain of 6 carbons, we have got an amine group here, and we've got an amine group there. So we've got 2 amine groups. The prefix for that is diamino. So we've got diamino, hexanoic acid. And finally, you just need to be a bit more specific and say where those amines are. So one of them is on carbon number 2, and one of them is on carbon number 6. So it's going to be 2, 6, diamino, hexanoic acid. So hopefully those examples are enough for you to see exactly how to name these amino acids. Now, this is the sort of thing you might be expected to be able to name in your exam. Amino acids can get a little bit more complicated in terms of their structure. So obviously this one's more complicated than this one's more complicated than this one. But you will not be asked to name anything that doesn't fit your normal UPAC rules. So you should be fine with anything that they give you in the exam. So next we're on to properties of amino acids. So looking at the diagrams of these structures that we've got here for alanine, leucine and lysine, you can see that we've got a wedged bond on the amine group in each case. So you can tell from that you're likely to have optical isomerism. And if you have a look, we've got a methyl group, carboxylic acid group, amine group and a hydrogen that you can't see. 4 different groups attached, so this is a chiral carbon. Same thing happens here, same thing happens here. So they are chiral. They do exhibit optical isomerism. And so there are 2 versions of each of the amino acids. And now these get denoted R and S, L and D, so different naming systems for them. You don't need to worry about how to name optical isomers as one-handed or the other. You just need to be aware that they do exist. So in biology, in our cells, we always use the L isomer and not the D isomer. So because their 3D arrangement is different from their other isomer, these fit with our molecules, with our biology, when they're in the L form and not when they're in the D form. So it's important that we get them in the right, as the right enantiomer. Now, it is just literally something to be aware of where they can pull something in that you've seen before in a different topic into a question about amino acids. So they could be asking you about this amino acid, they could ask you about the condensation polymerization that it undergoes and how it forms proteins and things like that. But they could also ask about the optical isomerism in terms of the fact that there are 2 and how would you figure out which one's which. So the rotation of plane polarized light. Just be aware that they can bring that into your question. Another property that amino acids have is that on one end they are acidic and on another end they are basic, because an amine is a basic group. So one compound that has both acidic properties and basic properties is described by the phrase amphoteric. So amphoteric denotes a compound that has both acidic and basic properties at the same time. So all amino acids are defined as amphoteric. So this is going to lead on to our next part of the discussion about amino acids. But if you think about the term acid, it means proton donor. So in the acidic group, each of these can donate this proton, release it as H+, and become a carboxylate end on there, OO, with a negative charge. So it's acidic. In terms of basic, what we mean is proton acceptor. So all of these groups, outlined in blue, can accept a proton to become NH3 with a plus. And that is going to lead on to our next property of amino acids. Okay, so amino acids, because they can act as acids and bases, quite often they don't exist like this, as we draw them usually, with an amine group and an acid group and no charges. Usually they are charged. So if you put an amino acid into acidic conditions, remember that means at low pH, because that tends to be how they write it in an exam. If you put this amino acid in low pH conditions, then what you will get is the following. So acidic conditions, low pH, means we have got H pluses available. And what will happen is your acid will stay as an acid, because there's plenty of H plus around for it to do so, while your amine group will become protonated. So it picks up an H plus, it acts as a base, and it gets a positive charge. So this is protonated on the amine group. So it becomes NH3 plus instead of NH2, and your acid stays as it was. In high pH conditions or basic conditions, we get something different happening. So here, in basic conditions, we end up with our amino group, unchanged, still NH2, but on our acid end, because it's a basic condition, in basic conditions, we end up deprotonating the acid. So the H plus comes off, this amino acid is deprotonated, and we lose that hydrogen. So in acidic conditions, your amino acid will have a positive charge because the amine group is protonated. In basic conditions, your amino acid will have a negative charge because it has been deprotonated at the carboxylic acid end. And if you're in between, if you're at a point where your amino acid does not have a charge at all, it exists as what we call a zwitterion. So I'll show you what that looks like now. Okay, so the point at which your amino acid is not charged at all is called the isoelectric point, and at this point you are deprotonated at the acid end and protonated at the amine end. So a zwitterion is a compound that has a positive ion on one end and a negative ion on the other end. So it's got both positive and negative charge, completely localised within the compound, but overall it means it does not have a charge. So this is called a zwitterion. And this only exists at what we call the isoelectric point, which is the point at which, in terms of pH, your amino acid does not have an overall charge. So you need to be able to draw any given amino acid in all three of these forms. So they may say, draw what it looks like at low pH, draw what it looks like under basic conditions, draw what it would look like at the isoelectric point, or draw the zwitterion for this particular amino acid. So you need to make sure that you can draw all of those. Okay, finally, the last little bit in this introduction to amino acids is how you can separate a mixture of amino acids. And you should be able to recognise this. This is chromatography. So to separate amino acids, we use thin layer chromatography, or TLC. So hopefully you recognise this process anyway from your GCSE or from your A-level topic on chemical analysis. But to separate amino acids, you dot a mixture of your amino acids along the bottom, along with some known amino acids as well. And you place this in a solvent. The solvent will take the soluble amino acids up the TLC plate, and they will come out at different distances depending on the solubility of the amino acid. Because all the amino acids have a different R group, and they're all got a different structure, they do all have different solubilities, so this works out as a good way of separating amino acids. So a very simple way of determining whether you've got a certain amino acid is to compare your mixture of amino acids with some known amino acids and see if they come out at the same distance above your baseline or your start line. Obviously, a slightly more precise way of doing it is to calculate what's known as the RF value, where you take the distance travelled by a spot and you divide it by the distance travelled by the solvent, and you give that dot an RF value. You then do it for the next one, the next one, and the next one. So you take the distance travelled by the spot, divided by the distance travelled by the solvent, and give that one an RF value. When RF values match, you know that you have a match between amino acids. Now, the last little thing you should be aware of is that amino acids are colourless. So when you carry out thin layer chromatography, you don't actually get all these lovely colours and you can't see where your spots are. So there's a couple of things you can do. You can either use a special TLC plate that when the amino acids travel up, if you were to place it under a UV lamp, your spots would glow. So you hold your TLC plate under the UV lamp, and underneath you take a pencil and you just draw around your spots, and then when you take it back out of the UV lamp, you can then calculate RF values from your pencil marks. So UV lamps is one way of doing it. The other way of doing it is shown here. In this one, they are using a locating agent. So this is what it looks like before, and this is what it looks like after. And the locating agent that we tend to use is a purple substance called ninhydrin. And ninhydrin is just a compound that you have in a spray bottle, and when you've finished your TLC and you've marked on where your solvent has got up to, you then hang up your TLC plate using like a peg, and you spray it from a spray bottle, and it will develop and it will give you these purple stains, and the ninhydrin sticks to your spots of amino acids. So make sure that you know how to carry out thin layer chromatography in the first place, and you can describe a decent method for it. Make sure that you are aware of how to calculate RF value and its importance in identifying amino acids. And finally, just make sure that you are aware they are colourless, and so you will either need a UV lamp or you will need a locating agent such as ninhydrin to identify your spots and to be able to then calculate the RF value. And that is all for amino acids. So we'll be moving on to proteins next. Thank you for watching.